Mycobacterium. It's a name of this special genus, Mycobacterium. The common features of Mycobacterium are okay. Here's the electron microscope picture, scanning electron microscope picture, and you can see they are rod-shaped, okay, slender, relatively thin and long, and they are aerobic, so they require oxygen, but they don't form spores, and they are not easily stained. Like the regular staining method, like the gram stain, it's not easy to stain the Mycobacterium. And to stain the Mycobacterium, there was a special design method called acid-fast staining. I guess that's the third lab you will do this, the acid-fast staining to stain the Mycobacterium. So basically, it's a staining method with a decolorization step, which can decolorize other bacteria but not Mycobacterium. I'll show you later. There are two major pathogens in this genus. One is the tuberculosis. We'll introduce this in detail. Another one is Mycobacterium leprae, which causes a disease called leprosy. Has anyone heard of this name before? Leprosy? No? Yeah? Yeah, leprosy is a disease influencing the patient's skin. It's invasive, it invades to the skin. And it also causes person-to-person spreading. Usually in the classroom, we don't introduce leprosy because we haven't seen this disease for decades. But it happened, okay, it happened yesterday. A doctor called me about this disease. They have a patient right now in the hospital. Doctors are very confident that this patient is having leprosy, which is very rare because they haven't seen this for a long time. But they, according to the symptoms, they suspect that the patient is having this leprosy disease. But they want to identify the bacteria in the patient's skin specimen. They collect the skin specimen, and they want to make sure that it's because of Mycobacterium leprae. And they ask me questions about the acid-fast staining to see whether they can find the bacteria. Hopefully, they can diagnose the patient as soon as possible because leprosy can spread person to person. So it's dangerous if it's true. Okay, we'll focus on Mycobacterium tuberculosis. Okay, this is a disease which is not common anymore, but still it's very infectious. It's everywhere. Even here in Zhengzhou University, in a student's dorm, several years ago, there was some active patient in a dorm, and all the students in the same room were isolated. And yeah, but it was okay. Finally, the student was rescued, and other students are okay. The Mycobacterium tuberculosis. The bacteria look okay here. It's thin and straight bacilli, especially collected from the patient's tissue. It looks like this. But if you culture the bacteria in the lab, they might show a variety of morphology. The morphology might change a little bit. And they are not stained readily because on the surface of the cell, it contains a lot of wax, lipid molecule. And that lipid molecule is extremely hydrophobic. And the dye we are using for like gram stain and other stain, the dyes are hydrophilic, which is water soluble, so that the dye cannot easily get into the cell. And to stain this bacteria, Mycobacterium tuberculosis, we use the acid-fast staining. So this is the method we use. Okay, the dye we use is called carbofusin. Okay, it's a red dye. We make a smear on a glass slide, then stain it with carbofusin to help the dye get into the cell. Usually we need to apply heat, okay, to heat up the slide a little bit so that the dye can get into the cell. Then after that, we decolorize the specimen by acid alcohol. So this is 95% alcohol containing 3% of acid, okay, hydrogen chloride acid. And this is a very strong decolorization agent which can remove the dye from other bacteria and also from the human cell, but not the Mycobacterium. The Mycobacterium is more resistant to this decolorization. After this, we counterstain with a blue dye, okay, so that after staining, the Mycobacterium will show red color, while other bacteria will show in blue color. This is the third lab you are going to do. Okay, this is the results of acid-fast staining. And you can see here in this picture, the red rods, the thin rods, are the Mycobacterium tuberculosis, while the green ones, like the small green ones, these are the other bacteria, while these big green dots are the nuclei of human cells. So this is a acid-fast staining. Okay, Mycobacterium, we can culture them in the lab. We can use the semi-synthetic agar medium, or we can use the egg medium. This one we call the Lowenstein-Jensen medium. I used to make this medium in the lab when I was in college at your age. It contains salts, glycerol, fresh egg or egg yolks, and potato flours. So at that time, I remember every a couple of weeks, I go to the supermarket to buy fresh eggs. Then I mix the egg yolk with glycerol and potato flour in the lab, mix it well. Then I add in a special dye called malachite green. This is a chemical. Malachite green dye into the medium. This is a dye which can inhibit other bacteria, but it cannot inhibit Mycobacterium tuberculosis. Mix it well, then we put the medium into the glass tube like this. It's still liquid. Then we put the liquid into the oven to bake it. In exactly the same way we make cakes at home. So after baking, it's solidified like this. Then we can culture the bacteria tuberculosis on the surface. So this is the solid media. We can also make the broth media to culture the bacteria in the broth. But the problem is, tuberculosis grows so slowly. It took at least 2 weeks to see the single colony. Sometimes we need to culture the bacteria for 4 weeks, a whole month to see the colony. Okay, the colony is like this. Okay, either rough or like this glistening, okay, greasy colony. It's because on the cell surface it contains a lot of lipid molecules, so it's greasy colony. Tuberculosis are obligate aerobes, requires oxygen, but their biochemical activity are not characteristic. So usually you don't have to run different type of biochemical experiments. They grow very slowly. The doubling time or the generation period we mentioned before is about 18 hours. Can anyone remember the generation time of E. coli? 20 minutes. Yeah, 20 minutes. And this doubling time is about 18 hours, so very slowly. Okay. And the bacteria is also resistant to a lot of factors, such as resistant to different dyes, like the malachite green. So that's why we can add this dye to the medium. It can inhibit other bacteria, but tuberculosis is resistant to it. It's resistant to penicillin, the antimicrobial drug. It's resistant to acid and bases, alkalis. So based on this, we can treat the sample, treat the specimen with either acid or alkali to eliminate other bacteria, eliminate contaminations, so that it will help us to culture tuberculosis with a higher chance. And it's also resistant to drying. The bacteria can survive for a long period of time in the dry sputum, the patient's sputum. The bacteria can survive in it for a long period. Here, long period means several months, so that it can be infectious and contagious for a long time. Tuberculosis does not produce endotoxin, they do not produce exotoxin, and also they do not produce invasive enzyme. Sounds like they are not dangerous, but actually tuberculosis is one of the most dangerous pathogens in the history. Before the invention of specific medicine, tuberculosis was named as a white cancer. It killed a lot of people in history. The most important virulence factor by tuberculosis are the different lipid molecules. They produce the lipid molecule as their virulence factor. But they also produce some proteins and polysaccharides as their virulence factor. Especially, they cause the delayed hypersensitivity. This is due to the production of proteins. And also the hypersensitivity due to the proteins. Here are the lipid molecules produced by tuberculosis. Okay, just let you know that you don't have to remember these details. Just keep in mind that the most important virulence factor are the lipids. You don't have to remember all those names. Tuberculosis infection, there are two types. One is the primary infection. The second type is the reactivation infection. For the primary infection, it's possible that if we inhale the bacteria into the lung, it can cause an acute exudative lesion and rapidly spread to the regional lymph nodes. The bacteria multiply in the lung, so that's the lesion. And in the lung, it can infect macrophages. Then the macrophages migrate to the regional lymph nodes with the bacteria inside them. So that's the primary infection. And usually, the infection site, the exudative lesion, can heal rapidly after the primary infection. And the lymph node, okay, once the bacteria get into the lymph node undergoes massive fasciation, which usually calcifies. the caseation and calcify, which makes the lymph node like in a cheese-like structure. Okay. And after the primary infection, the tuberculin test will become positive. Later, I'll introduce what is tuberculin test. A second type of infection is the reactivation infection, which is an endogenous infection caused by the tubercle bacilli, which survived after the primary infection. Okay, they survived in the primary lesion, and this reactivation infection can cause chronic tissue damage and the formation of tubercles, caseation, and fibrosis in the lung. So the lung is the primary site of infection. But it's also possible that tuberculosis can cause other infection in other organs, such as the bone, such as the intestinal tract, and it can cause lesions there. But the lung is a primary site of the infection. And this is the caseous tubercle. Okay. It looks like very similar to a piece of cheese, right? And the texture, the color, caseous tubercle. Immunity. Our immune system can fight against tuberculosis. And when the immune system functions, it can localize the tubercle bacilli and retard their multiplication, limit their spread, and reduce the lymphatic dissemination. But it also induces hypersensitivity, especially the delayed hypersensitivity reaction. That's due to the activation of T cells. Let me show you how to test the hypersensitivity. So this is the tuberculin test. Has anyone heard of tuberculin? Tuberculin was the protein component in the broth culture. So you culture the bacteria, tuberculosis, in broth for a long period of time, 6 weeks. And it produces protein molecule in the broth. Then you filter out the bacteria and the protein component in it. That's a tuberculin. And when we introduced Dr. Robert Koch, he worked on tuberculin for a long time, and he won Nobel Prize due to his work on tuberculin. But he worked on tuberculin. He wanted to use tuberculin as a vaccine, but it does not work. But we can use tuberculin for diagnosis. So what we do is that we concentrate the protein and inject the protein underneath the skin. Okay, if there is delay type hypersensitivity on the skin, you would see the swelling, the area of redness. So you know that in this person, there is delay type hypersensitivity. So this person might be infected by tuberculosis, or that the person received the BCG vaccine. Okay. And nowadays in the hospital, we are not using the old tuberculin anymore. We are using the purified protein derivative from tuberculin. So we purify the protein and use it as tuberculin. So do you have any idea whether you received the BCG vaccine when you were little? Did you? Yeah. Because in China, BCG vaccine is utilized, but it's not utilized in every country. In Western countries, BCG vaccine is not used. So if we receive the BCG vaccine, if we do the tuberculin test or the PPD test, it's possible that it will turn out positive or weak positive because we are immunized with the BCG vaccine. Okay, so this is the tuberculin test. Clinical findings after tuberculosis infection. The patient would suffer from fatigue, weakness, weight loss, fever, night sweat. They would suffer from vigorous cough and spitting of blood because of damage in the lung tissue. And without treatment, the patient usually would die. And that's why we call the tuberculosis the white cancer. And bloodstream dissemination can lead to the miliary tuberculosis. Bacteria get into the bloodstream, disseminate to other parts of the body because of miliary tuberculosis. Okay. Laboratory tests, that's the specimens include the fresh sputum and other fluids you can collect. And talking about the sputum, the sputum sometimes is not in liquid form. You need to liquefy it. So you can use this special amino acid, an acetyl L-cysteine, which can liquefy the sputum so that after that, we can treat it with sodium hydroxide, which is strong alkali, which can kill other bacteria in the specimen. Then we can neutralize the system with a buffer. This neutralized buffer is an acid buffer. Then we can concentrate by centrifuge to collect the bacteria. Then we can culture the bacteria, or we can stain the bacteria with the acid fast staining. Okay. For diagnosis, okay, acid fast staining is a special method for this. And to culture it, I just want to let you know that because the bacteria grows so slowly, it's not easy to culture it in the lab, especially from the patient specimen. Usually, it turns out false negative, okay? It contains bacteria, but you cannot culture it. Nowadays, we use DNA detection more and more. In the hospital, we can run a real-time PCR to detect the presence of a certain gene, and you can get the results in maybe a couple of hours. Okay, that's the medicine we are currently using to treat tuberculosis. Okay, one more thing. The problem right now is that tuberculosis is becoming more and more resistant to the current medicine. And that's why the doctors are a headache to this, because all the current medicine cannot kill the tuberculosis in the patient's body. So that's a big problem. So hopefully scientists could discover more and more new medicine to treat the bacteria. Okay, prevention and control. If the patient is actively having an infection, we should isolate the patient, cure them with the medicine we mentioned just now. And usually, the patient need to be isolated. Like in China, every city have one special hospital to take those patients with tuberculosis. So that's a better strategy right now. And to prevent infection, we can use the BCG vaccine. But we should know that BCG vaccine only works in the little kids. For you guys, adults, if you receive BCG vaccine, the effect is really poor. So it's not useful anymore to give adults with BCG vaccine. Okay, that's for Mycobacterium tuberculosis. So till now, we have finished talking about bacteriology. Next week, we'll talk about viruses. All right?